Industrial biotechnology processes comprises of different units. I told you that it comprises of 3 different units. One is your upstream processing, then your bioprocess where you have the bioreactor, and then you have downstream processing, and that is kind of separation process. Now, for the operation of any kind of industrial process, we required different materials. We have, as for example, first in the industrial biotechnological process, first we shall have to prepare the culture, and then that we consider as the inoculum, that is we call it seed, and this seed material we shall put it in the production fermenter first, because the seed material usually the size of the seed material is 5 to 10% of the production media. So, that cannot be produced in the lab. So, that is usually produced in the industrial scale. So, that is to be in a bigger reactor, and then this inoculum is supplied to the production fermenter for the getting the desired product. Now, I shall give you a small video where we try to demonstrate that biohydrogen production, because we have been working with this process for last couple of years, and we use this intervector clocky for the production of biohydrogen. There we are going to show you how we can prepare the culture, and how we prepare the inoculum, and then we use the immobilized whole cell system, because immobilized whole cell system is advantageous as compared to suspended cell, because you can hold the cell on the solid matrix and pass the substrate from the bottom of the reactor and take out the product from the top, you will get the product. Now, here we can immobilize cell in the bigger 800 milliliter reactor and continually pass your substrate media and get the hydrogen production. And here the product is hydrogen, and when hydrogen burn, it will give you the colorless flame, and it will give the water as a product. No way it will polluting the atmosphere. So, this video will give you kind of demonstration of the same.
Hydrogen as a promising alternative energy carrier can be produced in several ways, including environment-friendly methods such as biological systems. This pilot-scale study envisages microbial hydrogen production using fermentative bacteria Enterobacter cloacae IITBT08 in an immobilized whole cell bioreactor packed with eco-friendly and cheap packing material coconut coir. Any microbial fermentation starts with inoculum preparation. A pre-inoculum is first prepared from the preserved stock culture. After growing at 37 degrees Celsius overnight, it is added to bench scale continuous bioreactor, in which the actual inoculum is made using synthetic growth media. Sterilization is necessary to control the contamination by competitive bacteria and thereby improve the hydrogen productivity. The inoculum is then pumped into the hydrogen bioreactor containing sterilized buffer and carrier material. The construction of the hydrogen bioreactor consists of a jacketed reactor vessel, 800-liter working volume, a recycle tank, water seal, raw gas collector, automated logic control system, carbon dioxide absorber, and a final gas collector. Starvation stimulates the cells to immobilize on the carrier material during stabilization. Buffer is then drained out and sterilized media is pumped in using peristaltic pump. The bioreactor is initially operated in batch mode until hydrogen production peaks and then continuous flow of the media is started. Samples are collected at regular intervals for subsequent analysis. The gas produced is allowed to pass through carbon dioxide absorber before it is collected in the final gas collector. The gas produced is analyzed in gas chromatograph to determine the percentage composition. Gas produced is crudely analyzed for hydrogen content by burning in a flame.